So, dear audience of today's BIH online lecture, 7 days before Christmas Eve, it's my pleasure to introduce our speaker, Professor Matthias Lutolf from the Roche Institute for Translational Bioengineering and EPFL Lausanne. Matthias acts as scientific director of this institute and also as professor at the EPFL. It is safe to say that he is one of the world's expert on biomaterials and stem cell development, very successfully combining technological knowledge of various biomaterials with excellent stem cell biology. How comes? Well, he studied materials engineering at a famous university, the ETH in Zurich, and received his diploma there in engineering in 1999. Subsequently, he got a PhD in biomedical engineering, again at the ETH in 2003, and moved on to work, amongst others, as a postdoc at Stanford University. In 2007, he founded his own laboratory at the EPFL in Lausanne, where he was promoted to associate professor in 2014 and full professor in 2018, along with his directorship of the Institute of Bioengineering. In June 2021, so just a few months ago, Matthias Lutolf became scientific director of the newly established Roche Institute for Translational Bioengineering in Basel, Switzerland. His research focuses on the use of cutting-edge bioengineering strategies to build stem cell-based organoids with improved reproducibility and physiological relevance for basic research and also for real-world translational applications in drug discovery and development. Importantly, the discussion today will be moderated by another famous person, Professor Hans Schöler from the Max Planck Institute in Münster. Happy to have you here also, Hans. You know Hans Schöler is president of the German Stem Cell Network, the GSCN. But now, dear Matthias, we are proud to have you amongst us, of course, in the first place, and we are looking forward for you to give the BIH lecture on our virtual stage, which is now yours. Thank you.
Thank you so much, Professor Baum, for the very kind introduction. It's a great pleasure to be here. I also want to thank you, the audience, for spending your lunch break listening to me. I certainly appreciate that. So I would like to talk about the topic that has been a central focus in my lab at EPFL for the past at least 5 years, where we have tried to improve organoid technology using various engineering approaches. And I would like to give you 2 examples of how we really interface these fields to make organoid technology more potent for real-life applications. I think I don't need to introduce the topic of organoids to this audience because probably most of you are already using organoids in your research work. I think it's fair to say that this technology is really changing the way we can approach biology in the lab, and it excites many people also outside of basic science to try to see how we can use organoid models, for instance, for drug development and drug discovery, but ultimately also for regenerative medicine. So the field, I think, has been maybe pioneered by 2 pieces of work. One focused on adult stem cells, and in particular intestinal stem cells, by the lab of Hans Clevers in the Netherlands and Toshi Sato, who have really elegantly shown us how you can grow quite complex epithelial tissues from primary stem cells. And this model, to this date, remains one of the paradigmatic model systems used in many labs around the world. Yoshiki Sasai's lab, maybe around the same time, has really focused on using pluripotent stem cells to derive various neural tissue. And maybe the most famous example is that of these optic cup organoids that were generated in 3D cell culture from pluripotent stem cells. But now, in the past 10 years or so, we have seen many exciting developments by hundreds of labs around the world who are adding models multiple models to this rapidly growing list of organoids. So I think maybe one of the most compelling aspects of organoid biology and technology is the fact that, first of all, we can start to move into studying human biology. I think that's opens up so many exciting perspectives. The other, I think, very exciting aspects of the technology is that it presents a really interesting compromise between overly simplistic two-dimensional cell cultures that are used, of course, still a lot in many labs, and animal models, which of course are organisms and thereby extremely important, but they're also quite complex. And kind of, I would argue that organoids fall sort of in between these two extremes, where we have a certain level of complexity, but we can also start to use organoid at scale if we figure out how to derive assays. So we can design, high throughput systems, for instance, for discovery, for drug development, which makes the technology extremely interesting. Now, I'm personally, at the moment, very much interested in trying to see how organoid technology can be used for drug discovery and drug development. On this slide, you can basically see the state of the art in terms of how the industry is using human model systems for the various stages in the drug development process, from target identification to discovery, lead identification to basically assessment of safety and efficacy. And for these different stages, at the moment, animal models are primarily used, and there is also cell lines that are used, for instance, for discovery and lead identification. And now, of course, with the emergence of organoid technology, there is this vision that starts to excite people that we can start to use human model systems for drug discovery and drug development. So basically generate tissues, healthy and diseased tissues, from materials isolated from patients. If that's possible, if we can realize this vision, I think it really might be extremely impactful for the industry. So because ultimately will lead to the reduction of the attrition rate, which is tremendous at the moment. It will lead to saving of time and costs, increase the number of new targets, and also reduce animals and have a better way to select patient cohorts for clinical trials. This is the vision, and I think we have a long way to go to realize it. And that entails, I think, solving some of the significant translational challenges that we have now identified in the field. And here I'm summarizing basically what I consider to be some of the main obstacles in the field that should be addressed, and engineering, I think, can play an important role in this task. So by and large, I would argue that stem cell development, the generation of organoids, is still difficult to control and results in quite a striking heterogeneity in terms of the types of tissues, the morphologies, the phenotypes, functional characteristics of tissues that are generated. Organoids are generally also very small. So even if we use bioreactor technology, we can't grow organoids larger than a few millimeters in size. Organoids, especially those derived from epithelial stem cell types, lack an in vivo-like anatomy. So they basically result in closed cystic structures instead of openly accessible tubular structures. And that might actually be very important for many real-life applications. And maybe most importantly, organoids are very simplistic in terms of their tissue composition, if you look at the entire organ. So typically, growing epithelial stem cells, we only capture one type of tissue, and a very important tissue, of course, but only one type. So we're lacking immune system components, vasculature, stroma, innervation, etc. So there's a long way to go here to really capture the organ level, and not even mentioning the organism level complexity and function of in these existing in vitro models. I will be focusing in my presentation on this model here because I think it's a very powerful one, and it's one that's easily tractable, and we can access it very easily in the lab. So here Hans Clevers was basically demonstrating with Toshi Sato that you can isolate even a single stem cell and grow it in a 3D culture system, and you end up after a week or two with tissues that capture really the structural features and functional features of the native epithelium, where you have cryptomains with stem cells and panel cells, the niche cells, but also villus-like domains, which essentially comprise the key differentiated cell type of the native epithelium. This was a groundbreaking advance for the field, for sure. Now, if you grow these organoids in your lab, you could get something like this. This is an example from one of my PhD students who works with these organoids all the time. So you can see right away that the heterogeneity in these cultures is very striking. So you have organoids with different shapes, with different numbers of crypts, different sizes. And this is really, I would say, is the typical of most organoid cultures at the moment. And what we have asked in the lab a long time ago actually is whether the reason for this heterogeneity, or one of the reasons for this heterogeneity, might be that we actually don't have any predefined patterning instructions in these cultures, and we also don't have any physical boundary conditions, physical constraints, that the lack of which might actually result in more poorly controlled morphogenesis. And now, if that were true, we considered that engineering approaches to bring back some of those instructions, extrinsic influences for patterning, might be the way to go to make organoid development more reproducible, and also start to grow tissues with defined shape and size. And I want to illustrate this concept by two vignettes that we have been working on in the lab in the last few years. The first one is focused on a technology called 3D bioprinting, very well established technology, which we have used to try to scale, to push the micro scale self-organization process that leads to patterning to the centimeter scale. So a scale that could become relevant for human organs at some point. And in a second topic, I would like to focus on a different technology where we have microfabricated biomaterial scaffolds to be able to constrain morphogenesis to defined physical boundaries. So start to control the shape and size of organoids for the first time. So let me start with the 3D bioprinting approach that we have developed. Now, this is obviously a very established field. There are many labs around the world who focus and have developed very sophisticated hardware, new printers, but also so-called bioinks that allow you to really control very nicely how cells are positioned in 3D space. I think one of the major challenges in this field has been that it was very difficult to capture the sort of natural self-organization characteristics that organoids have in these bioprinted platforms. And so what we have basically done here is we said we're only going to focus on biomaterials that already allow organoids to be developed. So we don't engineer our own bioink. We just start from something that already works. And then we basically simply used an existing microscope that had a very precise XYZ stage to move around the position of a dispensing unit of a syringe-based extrusion system, such that we could deposit into a very biological 3D environment different cell types that could form organoids. And so here you can see how this looks like. On the left, the top corner, you can see our prototype dispensing unit, which is simply a glass capillary that my student fabricated himself, which has a diameter at the tip of about 70, typically about 70 micrometer, which allows us to basically deposit cells at very high concentration into this matrix shell before it actually fully polymerizes. Here in this movie, you can see how cells are deposited in 3D in a solution of 3D viscous solution. And here you can see various parameters that allow us to control this 3D printing. So we have the nozzle size, we have the speed of the stage, as well as the flow rate of the syringe pump that we're using. Now, in the simplest configuration, when you do this with intestinal stem cells and you grow them on the organoid formation condition, we could, right from the beginning, get a very nice sort of a line of cells that reorganizes into an epithelial tissue. And when exposed to organoid culture condition, we could see that these cells start to bud out and actually form new crypts, which was really beautiful to see. In this high magnification time lapse, you can see here that the cells in dark are basically the penna cells that are the organizers that generate the crypts and that maintain the stem cells in their native niche. So this simple approach that we developed essentially allowed us to generate fully patterned centimeter scale intestinal tubes. So basically scaling the organoid development process from the size of a few hundred of microns to the centimeter scale. And we find that this process is very reproducible. Here you see multiple repeats, different experiments. And the key parameter to control organoid or tissue formation is really the density of cells that you print in the beginning. You need to push this concentration to very high numbers, to millions of cells and tens of millions of cells, in order to get robust patterning and tissue formation. On the right side on this slide, you can basically see that we get very nice crypt structures with here cells, the stem and progenitor cells labeled by SOX9. And here you can see basically the penna cells in the crypt labeled by lysozyme. And we can see that we have villous domains as well with enterocytes. And here that the proliferating cells are really basically located in the crypt structure. And we have evidence for all the key differentiated cell types in these bioprinted constructs. We were interested in trying to see whether these tissues could respond to physiological stimuli as you would expect, as it happens in vivo also. So we use different assays. For instance, here we use carbamylcholine to expose the tissues to this compound, which basically releases the lysozyme granules from the penna cells. And in this time lapse, you can see that this is a very rapid response, and these granules are basically expelled from the penna cells. On the right side, you can see that exposure to forskolin, which activates the CFTR channel, basically results in a tissue scale response and a physiological response that you would expect, similar to organoids, basically. So these tissues really behave in a physiological manner. And we also asked whether this approach of bioprinting epithelial stem cells in these 3D matrices could be adopted for various other types of stem cells. And the answer is yes. So here we have examples of human colon stem progenitor cells, mouse, as well as stomach cells. And in each case, we can basically generate a larger tubular epithelial structure. Now, I think one of the perhaps unique aspects of bioprinting is that you can start to control in time and space how different cell types are positioned in 3D. And that is exciting, because you could think about generating more complex type of configurations where you have multiple types of cells deposited that form multiple tissues and tissue boundaries. And you can also think about, studying interactions of different cell types and tissue types to increase multi-tissue complexity in these 3D cultures. Here's, for instance, one early example from the lab where you have deposited stem cells, intestinal stem cells, next to myofibroblasts isolated from the same tissues, and you can see that these cells start to crosstalk. They start to interact with each other, and actually the myofibroblasts influence also how the stem cells are forming the epithelium. And perhaps in the most interesting application of this technology, we focused on trying to generate two different regions in the gastrointestinal tract. So we used stomach stem cells and small intestinal stem cells, each labeled with a different fluorescent reporter. And what we could see in these experiments is that both stem cells basically generated their respective epithelial tissue, and that the cells this hybrid tissue maintained the expression of key markers of the stomach and the intestine, suggesting that it is possible to really start to rebuild this complexity of such a organ system, along an anterior-posterior axis, for instance. And also, whether it would be possible to find and to generate boundaries between different tissue types, which is something that I think is very hard to explore in vivo. And then finally, we are also interested in trying to see whether the lumen in these long, centimeter scale tissues could become accessible by, for instance, fluid transport to remove cells that have been undergone apoptosis, or to add bacteria or viruses, etc. And it's possible to do that, but we find that it's very, very difficult. So it's in this current configuration of the technology, this is not something that you can easily use for real life applications. And that has sort of been the reason for us to start to look into something that's perhaps more robust, where we used microfabrication of hydrogels to see whether we can generate these engineered organoids. And this is what I'm going to talk about now. So in this work, we have basically used microfluidic technology to generate a microchip where you have, access to a tubing system for perfusions. But very importantly, again, we have basically used a highly permissive organoid biomaterial that allows organoid derivation, and we have placed that in the middle, in the center, in this chamber here. Here you can see a top view, and here a side view. I want to focus now on this sort of central component, which is really the key one, where organoid development takes place. So here you can see that we basically used, we generated basically a perfusible microchannel that connects two liquid reservoirs with each other. So you can have fluid transport. And we generated this microchannel by using laser ablation, so very precise micro fabrication process. And we also generated these little crypt-shaped cavities next to the central channel here. So the hydrogel was basically should act as a basement membrane-like substrate to allow stem cells to adhere and get polarized to form an epithelium, but at the same time also kind of replicate the micro topography of the native small intestine in a simplified manner, of course. And here is what happens when you add primary stem cells from the small intestine. They're very nicely adhered. They start to proliferate because we expose them to we activate the Wnt signaling, so they really proliferate very rapidly. And over the course of just about 2 days, these cells have grown so much that they cover the entire inner surface of this scaffold that we have provided, and they form really a beautiful epithelium, a columnar epithelium after about 2 to 3 days. On this side, you can see what the difference is between a classical organoid culture, where cells are able to freely grow in 3D and acquire different shapes and sizes, compared to this sort of scaffold guided organoid derivation approach that we developed, where you can immediately see that we are able to control how this epithelium is formed, what type of shape it has, what type of size it has. Now in this image, you basically see a cross section through the entire epithelium using confocal microscopy, where we have labeled E-cadherin as well as the nuclei. And that, I think, very nicely illustrates how perfectly organized these tissues are, and that they remain open. So we have an opening on both sides such that we can deliver fluid through these epithelia. And here you can see where we have perfused the tubes with Fitzy labeled dextrane, and you can see that the epithelium is a very good barrier. So there is no permeation of the macromolecules through the epithelium. Now, one of the, I think, most exciting moments in this project came when we changed the culture conditions from expansion of stem cells to generate an epithelium to organoid patterning, to basically remove wind from the cultures and basically allow the stem cells to differentiate also. What we saw is that, you know, in 1 to 2 days, we basically had generated a highly stereotypical patterning where the stem cells were located in these crypt cavities, just like the Panna cells, the niche. And while the differentiated cells shown here, the enterocytes or goblet cells, as well as endocrine cells, were basically located in the central villus-like domains. This suggested to us that the geometry of the scaffold itself influenced the patterning process, because we exposed all the domains of the tissues to the same culture environment. We also ask ourselves, you know, what's the cell type composition of these epithelia? And we compared it to classical organoids. And by single cell sequencing, we could identify a couple of very interesting differences. For instance, we saw that in classical organoids, we very rarely get enteroendocrine cells, very important cell types for hormone secretion that are difficult to generate in decent numbers in classical organoids. But in these engineered tissues, we have a very sizable proportion of these cell types. And we also found that we had cells that we in our hands did not appear in current cultures, that are called M cells, microfold cells that basically take up antigen from the lumen and interact with lymphoid structures like Peyer's patches, where you have immune cells that really play a key role in the immune regulation of the intestine. These cells were also quite abundant in these engineered tissues. And we have other cell types that could not be detected in current cultures. So I want to take just a few minutes now to focus on this question of whether and how tissue geometry could control organoid patterning in this system, because it was something that we really considered as quite surprising. In order to address this question, we actually reverted to an even simpler model system, where we generated small cavities of defined shapes on a hydrogel surface using elastomeric stamp, like a micro-patterning approach. We then added the stem cells that were trapped in these cavities, and we closed these cavities with another layer of hydrogel, such that we really have confined the stem cells and forced them to grow in very well shaped and defined microcavities, arrays of microcavities. And we then used high throughput image analysis tools to extract information on where certain cell types were located in these small microtissues. Here are some examples. So here we have used circular arrays of circular patterns, where you see the epithelium very nicely adapt to these micro tissues. Here is another one which we have studied a lot, because this pattern here basically mimics the geometry of the native crypt in the small intestine. And you can again see that we get a very nice polarized microtissue epithelia that adopt to this particular configuration. Now, this is very important here. What we see is that when we grow these organoids in this confined microtissue configuration, you see that the patterning process again becomes highly stereotypical. And we have analyzed this in a quantitative manner using segmentation tools to basically generate frequency maps of certain cell types in these tissues. And we looked at hundreds of microtissues for each cell type. And what we can see is that the stem cells are basically located at the end of these tubular shapes. So we have really this high preference for the appearance of these cells here. Same is true for Panus cells, while the differentiated cells form a domain in the central regions of these microtissues. And we ask ourselves, what's the potential mechanism that can explain this geometry-controlled patterning in this system? Now, the first kind of change in cell behavior that we can detect is basically related to the shape of the cells and the packing of the cells. What we see is that very early on, cells that are located in these tip regions, in the curved regions, are basically packed together very closely. We can measure that by large differences in internuclear distance. And we also notice that the shape of the cells is very different. So when they're sitting on the side, you have much larger cells, higher surface area, while when they're packed together, you have a much smaller surface area, and the cells are really wedge-shaped. And this difference in cell shape basically translates into differences in the appearance of nuclear YAP. So the hippo downstream effector, the hypopathway YAP, a well-known mechano sensing and transducing regulator, basically is differentially expressed as a function of the shape of the tissue. And what we find here is that cell spreading and YAP activity are necessary for the patterning, because if we disrupt it by using, for instance, myosin inhibition with slabistatin, or by inhibiting YAP or activating YAP, in this case, the stereotypical patterning gets completely disrupted. And then, of course, we ask the question how this early mechanical pattern of nuclear YAP translates to differences in cell fate decision making and the appearance of crypt regions in these tissues. And here we basically were able to, luckily, basically, Prisca Liberali's lab had identified a mechanism that involved basically had differences in nuclear YAP as the key early event for symmetry breaking in these tissues that goes through a lateral inhibition process. And we were able to see that in these sort of engineered tissues, the same mechanism basically takes place, and that these differences in the nuclear YAP basically results in the controlled appearance of the first Panus cells in these tissues. And the Panus cells is very well known for basically secreting wind and generating the crypt and stabilizing it. So, you know, for those of you who are interested, you know, this is what we consider as a current working model of this geometry controlled patterning in this system, where you have first a mechanical patterning process that's followed by a spatially controlled symmetry breaking process in these tissues that results in this stereotypical patterning. Now, in the conventional organoids, a similar process might happen, but it occurs stochastically or random. So you cannot predict where a future crypt will be generated in a typical 3D cell culture. So with that, I close the bracket here, and I want now to move more towards tissue biology and try to see whether we can use these engineered models to look into some aspects of tissue dynamic, in particular homeostasis and also regeneration. I think one of the very neat aspects of this technology is that it's this open accessibility of the intestinal organoids basically allows us to continuously remove cells that are shed into the lumen. And there's a lot of shedding going on because these tissues turn over and completely every 4 to 5 days, actually. And so when you stop the flow, the perfusion through these tubular epithelia, what you can see is that a lot of cells get accumulated in the middle, and you start to see that this becomes more and more cloudy. But if you start to turn on the pump again, you push out basically almost a plug of dead cells from the lumen. What this suggests to us is that if the ability to continuously remove cells that are shed into the lumen basically allows to generate a balance between cells that are born in the crypt structures and cells that die and get shed into the lumen. And this, I think, allows us to generate in vitro homeostasis, which means that we can grow these tissues for very long periods of time without passaging. We can just continuously empty the lumen and get rid of the cells that have been turned over. And this remains a very stable system. So even after a month in culture, we still have stem cells located in these crypt cavities. And that's because we have this accessible lumen here. Now, what happens when you damage these intestinal epithelia, for instance, by gamma radiation? This leads to a massive damages in the epithelium. So entire fields of cells are basically disappearing. There's apoptosis of the cells because you have killed proliferating cells. But after a couple of days later, there are cells that are competent to regenerate, and they completely rebuild a patterned intestinal epithelia with stem cells located in the crypt. So these engineered versions of organoids have a remarkable ability to regenerate, which is, I think, an aspect that's very hard to model in classical cultures. Now, one of the, I think, outstanding features here was whether this technology, which we have developed for the mouse models, is applicable to human stem cells, to look into human tissue biology. And I want to first actually talk, before I come to the human models, I want to talk about some, I think, real-life applications which are very easily accessible because the lumen can be accessed. And so one application was basically in modeling infection, for instance, by parasites or by viruses or bacteria. And so here in this first application, we looked at a parasite called Cryptosporidium parvum, which really is represents a major medical problem, leads to, in some cases, deadly diarrhea in children and immunocompromised patients. And the question in this field has been, how does this parasite basically undergo its life cycle? And there are many open questions in the field. And the challenge is to have a suitable model that allows you to capture the entire life cycle of the parasite. And so we collaborated with experts focusing on crypto. And we basically, had the first question that we ask is whether this parasite can undergo its entire life cycle, and which is very complicated, involves sexual and asexual stages. And according to the experts, this parasite can very nice, because we can grow the tissue stably for long periods of time, the entire life cycle can be basically regenerated in these tissues. And we find that this happens even over the course of an entire month, where we can continuously see the emergence of the oocysts, which are the spore size of the parasites, which you can easily label, and you can simply count how many oocysts are generated in these tissues over these long periods of time. So here we have a system that I think presents a very interesting model to start to look into the biology of these parasites. Now, is this now applicable to human stem cells as well? The answer is yes, but it took us quite a while to figure out how to do this, because the human organoids, they're not so easy to grow. They, in some cases, grow much slower. and they required somewhat changing the parameters of these scaffolds in terms of stiffness, matrix composition. But I'm happy to say that we managed to generate organoids derived from human stem cells. Here is an example of the colon, a human basically mini colon that also have some cell turnover. And they also have a certain level of patterning where we see proliferating cells in these crypt cavities, but not in the central regions in the villus domain. And very excitingly, we actually see a highly abundant population of goblet cells that secretes mucus. And you can see that when the tubes are emptied, you can see this highly viscous phase that emerges, which represents mucus that's secreted by the goblet cells. We have applied this concept to a number of different human systems as well. For instance, here is an example of the bladder, where we generate the very nice stratified epithelium from bladder progenitor cells. And that's in collaboration with a microbiologist here at EPFL. And this is, again, a very reproducible, very robust approach that can be used very easily. And in this case, we even find that we can control the pressure in the lumen to mimic this sort of filling and emptying of the bladder, to also introduce some aspects of mechanical stimulation in these engineered organoids, which I think is something that's also quite lacking in the field right now. Now, with these models, you can quite easily construct co-culture systems where one can start to model infection processes. So here we have, for instance, introduced these uropathogenic E. coli, UPEC, which also are a major medical issue. And this system now allows us to really study infection and persistence of some of these bacteria after antibiotic treatments. These are some examples. Now, in the last 2 minutes, I want to give you a perspective on where we are going with this technology. One really major issue that we all have in the field, I think, is how we go from a simple sort of mono tissue to a multi-tissue system that really mimics organ biology. And we think this approach that we developed can be quite suitable to move in that direction, because this hydrogel matrix that's positioned in the microchip in the center can be colonized with other cell types of the organ, for instance, immune cells or vasculature. And that's what we're currently working on in the lab. So here you see an example where we have introduced macrophages in the surrounding extracellular matrix of this intestinal epithelium. And we find that these macrophages are really activated and start to interact with the intestinal epithelium. They migrate toward it, and they also really tightly interact and form processes to penetrate the epithelium. And we have evidence by time-lapse microscopy that these cells are even taking up apoptotic particles, really undergoing phagocytosis, which is, I think, quite nice and shows that we can grow these immune cell types and capture them in a functional state. And finally, this is also an outlook where we are right now, is really to try to see whether you can bring in, other perfusible tissues like a vasculature network that could be positioned next to the epithelium to start to look at the crosstalk between these different compartments, and even have, stromal cell types like myofibroblasts embedded in the matrix. So we have 3 different cell types, and they can be maintained stably in these cultures. This is definitely, I think, where we're going now, and we're trying to see whether we can capture these tissues in diseased states that require multi-tissue interactions, in particular with an immune system. With that, I'd like to stop here, and I want to, reserve some time for the questions. I'm extremely grateful for the talented people who have done the work that I presented, in particular Mike Nikolaev, who has developed this tubular organoid model. Olga is pushing this now more into the human aspects. Nicola Brogio has developed single-cell sequencing. Tonio and Moritz have developed bioprinting work together with Jonathan, who was a master student in the lab. And Nicole Czerewski has really developed our micro-patterning and has pioneered actually the organoid work in my lab when he joined a long time ago. And I'm also grateful for Hans Klevers for the work on the cryptosporidium, which was very interesting work. Thank you so much for your interest.
So, Matthias, thank you so much for this fascinating, amazing talk. So these structures are so you can look at them for hours. It's really beautiful. So for all, now please, it's time to put in your questions. We already have some questions actually. First one from Georg Duder. And what he writes is, hi Ludovic, thanks a lot for a fascinating talk. The engineered organoids you showed have distinct 3D, it's moving around, 3D geometrical patterns, e.g. like radii. Have you observed influences by the radii that you selected in your model systems? Many greetings, Georg Duder.
Yeah, this parameter plays a very important role. I mean, maybe the major geometrical parameter is the diameter of the cavity. So you really have only a relatively small window that's close to what you can measure in vivo in the small intestine that results in the emergence of stable crypts. So if you're going below that, and this is typically 60 to 70 micrometer diameter. If you go down to 50 or lower, there is not even a crypt that can be generated. If you go above about, let's say, 80, 90 micrometer, this crypt is also not stable, and the cells or the tissue try to basically generate two crypts. So you have in this system a very tight size control that I think that still opens up interesting unaddressed questions in terms of how that's regulated. But it plays a big role for engineering such a system, that you really hit the sweet spot that leads to a stable pattern.
Great. The second question is by Daniel Besser. Matthias, excellent talk. What is the origin of the human cells? Could you get the organoids from human embryonic stem cells? And does it work with mouse embryonic stem cells?
Yeah, so we have focused a lot here on adult stem cells that we can get from clinical collaborator and now commercial sources as well, where you can just, there are biobanks that can be accessed, and you can test many lines from different patients. This is what we typically do, and of course, as you say, there's also this very interesting approach of starting with pluripotent stem cells. The challenge there is how do you get maturity? How do you push the cells to go into a state that captures the adult? I think it's a general problem in the field, especially obvious in the case of the brain models, but also in the case of intestine, to generate a stable crypt-villus axis from pluripotent stem cells has remained a major challenge. And we are collaborating with groups who focus on this to see whether this technology can actually help to promote maturation in the system, because you basically provide a certain shape that might have an influence on that process as well. But I'm with you in terms of exploiting also pluripotent stem cell as a source. I think it's very interesting.
So the third question is from Katharina Scheibner. And here I'm not really sure if this is like a repetition what Daniel was asking, if you're wondering if you also generated human stem cell derived crypt organoids. I guess that is human embryonic stem cells, so what you already answered, and the and otherwise you have talked about human adult stem cells. So I guess, but there's a second part to the question, and if I'm wrong, please repeat your question, Katharina. And the second part is, and does the cell seeding density also play a role for the patterning of your intestinal organoids?
Yeah, I think, yeah, it does. It does. I think the process is definitely, you can make it more efficient if you increase the seeding density even in this configuration, because there is crosstalk between cells, and you generate a nice epithelium much faster if you don't have only very few cells in the system. So it's also, in general, I think it's a parameter that affects everything, almost everything we do in the labs with organoids. And you can look at processes like organoid formation efficiency, and each time this parameter seems to play a role also in this case here. Yes.
Oh. So I'm not going to repeat again and again that the talk was great, because everybody starts how great the talk was. So
don't worry.
okay, and I'm interested in your opinion on how comparable is FABP and valine as markers for intestinal enterocytes.
Yeah, that is a good question, and I have to be careful in what I'm saying. Because we have been struggling quite a bit with markers, and FABP has been one very good antibody that we can use. It just works great, and we don't know how it functionally compares to valine and other markers. I think this is something I can't answer. I think there are better people, maybe even in the audience, who know that. Yeah.
And the next one, did you try to evaluate the functional aspect of this organoid system as an enzyme secretion profile, for example?
Yeah, we have done part of it, not completely. We have, for instance, looked at the function of peptidases that are sitting on these microvilli on the enterocytes, and there are some very good biochemical assays where you can just measure labeled amino acids, and you can look at how well these peptidases are processing basically food, and that looks very nice. Looks like they really have, these cells are functional. But what we haven't done, and I really want to do, is to look at the function of enteroendocrine cells. I think there's lots of interesting biology and questions in regard to those cells, and whether one can reconstruct aspect of hormonal regulation using organoids. And I think the beauty of this approach is that you really have control over how you deliver stuff. So you can measure, you can deliver on the apical side and measure responses on the basal side of the tissue very easily. And I think we have only scratched the surface in terms of what is possible when we move into physiology and really these types of questions that you're asking. Yes.
Then the next question, it seems that the 3D printed mouse human colon cannot form crypts. Do you know what the reason for that is?
Yeah, we tried to understand what's happening. So one issue, a major difference between the mouse and the human, is that in the mouse, in the small intestine, you have lots of Paneth cells, and those Paneth cells are basically acting as a niche. When you grow human intestinal organoids, you lack a decent, sizable proportion of Paneth cells, so you don't generate the fully patterned crypt-villus axis in these human organoids. And I think what is needed now is to identify the stromal, the mesenchymal niche cells that could deliver the morphogens. I think in the human it might be just the way to go, because it is very different from the mouse model. In the colon, you don't have a lot of, or no Paneth cells at all. So there we really rely on building sort of a cellular niche that stems from the mesenchyme. So I think these co-cultures with mesenchymal cells and epithelial cells, I think in the human system might become very important. It's a topic that's quite pursued by several groups in the field right now.
Mm-hmm. Before the next question, I also, before everything is over, I also wanted to know the cells that were shedded off into the lumen, you said are dead cells. Are they apoptotic cells? And because when you did the radiation experiment, it looked like you get the reculturing. is this not by the shedded cells? Are they really dead? Are they apoptotic or just dead cells?
Yeah, this is incredible actually, because when we do time-lapse imaging, we can use live markers, dyes for caspase 3 and 7. So we actually see quite nicely when cells become apoptotic. And what's interesting is that cells get extruded from the epithelium before they are still alive, before they start to express apoptosis markers. So it's simply that they get pushed out, and then the cells become apoptotic because they lack cell-cell adhesion interaction. So it's like you would expect, I think intuitively, that okay, cells die and then they get pushed out. But at least in this in vitro model that we have developed, it's the other way around. Is that first they get physically pushed out, probably because of a lot of forces that are acting when the tissue proliferates, and then they die. So in the case of irradiation, it's different. It's like you kill cells that are like probably mainly transit amplifying cells that proliferate a lot. And when you lose these cells, the tissue turnover is halted. It's like interrupted, and you create a big gap. So the cells that have been irradiated, they undergo, I mean, they die. And then you have big defects that can be then repaired after some cells that were mainly not affected by the radiation can basically migrate in and proliferate to rebuild the epithelium.
So you don't think that the shedded cells are rescued by the irradiated areas, that they are kind of, before they die, they kind of build a new crypt?
It's an open question. We think that some cells in the crypts or close to the crypts might not be. We wouldn't argue that you have like cells that are quiescent or they're not like in G0 that then can be activated and take over, you know, the stem cell function in the crypt. I mean, that's a bit too much to ask for, but there might also be some plasticity in that system, right? In that, I mean, there seems to be, based on in vivo studies, a lot of plasticity that even if you ablate, I think all, you know, stem cells, some other cell types can acquire stemness properties in this system, in mice at least.
Okay, so there are 2 more questions, and then we're going to have a 10-minute break, and then we're going to have meet the speaker. So all the questions, I think there are lots of questions still open there. So 2 more. That is one question. Since you're reconstituted organoids with macrophages, since you're reconstituted with macrophages, how important do you think that the co-cultured immunity is for the same donor, genetic?
Yeah, that will be very. I think to rebuild, to generate sort of immune competent tissues, we will have to be autologous. I think because otherwise there might be an immune response because of the different molecules on the surface of the cells. That clearly is, I think, where we have to go. That we need to build autologous models that are derived, where we derive organoids, but also T cells, macrophages from that same individual, because only then can you really start to kind of have native immune responses, right? But I think there is progress in terms of generating autologous models of higher complexity.
Okay, so the last question now, and then Francesca and all the others, please ask your questions then in the meet the speaker. Okay, the last question is, did you make the hardware equipment scaffold by yourselves, or could one buy one? So there's somebody apparently that you influence who wants to do this.
Yes. The chip technology is not yet commercially available. We have licensed the IP to a spin-off from my lab, and they're developing sort of a first version of these tubular organoids. In terms of the biomaterials, we were very disappointing for myself, because we have been working a lot on trying to replace matrix gel, and we still do. But we have been building everything on the poorly controlled materials like matrix gel and collagen. You know, we are used, because we were not, this was not part of the main purpose here, but I think it will be possible to work with chemically defined materials to build similar systems, and that's definitely an important step towards building a commercial system that can be disseminated. Yeah.
So thank you. As you can see, you sparked quite some interest. So there will be many more questions. So now I'm going to hand over to Daniel, who's going to be the one who is guiding everybody through the Meet the Speakers session. Thank you so much again, Matthias. Great.
Thank you, Hans.
And good luck with your endeavors since the summer.
Yeah, thank you very much.
Thank you so much, Matthias, for excellent presentation. We have a lot of people congratulating you, including Hans. And there was one real quick question, maybe we can do this, from Francesca, about the shape changes, for instance, to other kind of tissues, for the brain or for ventricles. Maybe you quickly, briefly answer this, but we will have definitely much more time to talk after a 10-minute bio break, and then we meet at 1:10. But maybe you can real quick say something about Francesca.
Yeah, Francesca, yes, I think so. I think we need to. I think the concept might play a role even in non-epithelial, like not only for the gastrointestinal tract. I mean, we have done now quite a lot of work on the respiratory system with lung, airway, liver as well, and I think similar ideas might be implementable with even brain tissues, where you need to generate the neuroepithelial sort of starting tissue that can then be further patterned, and I think to generate that in a robust way, shape can be one powerful way to go, I would argue. But we haven't done anything along those lines, but I think it's a really cool idea to pursue.
Thanks, Matthias. So see you in 6 minutes. Is that okay?
Okay.
10:10. See you then.
See you later. Cheers.